topic is that novel p53 independent non-cytotoxic therapy for small cell lung cancer. So what I will be talking today, so I outlined that I will be talking a little bit of cancer and lung cancer, what are those and how important lung cancer is, and then p53 loss and resistance of the small cell lung cancer to cytotoxic treatment. By telling that, what I mean to say, our standard treatment like chemotherapy, radiation therapy, those are the cytotoxic treatments. That's the one we are treating the patient right now. And then small cell lung cancer that are committed into neuroendocrine lineage and then maturation arrest. So this is our basis of the new drug development. So that we are focusing on to reprogram this process. So that's why I will be talking little bit details about that. Then how this mechanism happen. So there is, like, as we all know, the transcription factor, translational regulation, gene expression, and there is lot of co-regulator like co-repressor and co-activators. And I will be talking over there in cancer cell, co-repressor amplified and co-activator deleted. So that turns into the maturation arrest. And after that, I will be showing that if we target the co-repressor, which is amplified in cancer, in vitro and in vivo, we can fix this problem, the cancerous proliferation, and we can make those cells to differentiate as it was destined to. And next step, because my current mentor is a physician, so he is taking all these findings to the clinical trial. So, and anytime, anywhere you guys have question, please stop me. I will explain. The cancer is the second leading cause of death globally after the cardiovascular disease. And it is one in every six patient, sixth death in the overall world is from the cancer. And the cancer basically, it's like normal cells which is having the abnormal cell growth or proliferation. I mean, where it stops, it's not stopping, it is going beyond that. That's in simple way, if I will tell that. So how it happens, it's just normal cells in our body that transformed into tumor cells, and at that time it is a complex process. I will get through it. And which that changes, it made that uncontrolled growth. In normal cells, what we see, like it grows for some time and then it restricted. So that's why our multicellular organism is, we are in a particular shape and organ, everything properly functioning. And I just put a graph from the World Health Organization, and you can see, although the breast cancer, you will see the event is more, I mean, we see a lot of breast cancer patient, but death-wise, if you see, lung cancer is the leading cause of cancer-related death. So it's very important because of that. And why it is happening, I am going to talk about that. So this is the same one, I just put it in a bar graph way so that it will be easier to see that. So see the breast cancer, it is event is incidence, how many people are suffering, you see that, but the relative to that, death is not that much. At the same time, lung cancer, see the how many people are suffering and death, almost everyone. So now lung cancer, there are two types of lung cancer. One is non-small cell lung cancer, which is the majority of the lung cancer, and the small cell lung cancer. Although non-small cell lung cancer is the major part, but the small cell lung cancer is the worst one. When it is detected, almost not curable. So almost everyone is dying. So this small cell lung cancer is the most malignant subtype of lung cancer and characterized by the aggressive growth, and it early onset it get metastasized when it spreaded beyond the lung, and its rapid development of chemorefractory disease, which means it became resistant to chemotherapy and radiation therapy. And on top of that, 70% people, when they are diagnosed with the small cell lung cancer, already that is spreaded beyond the lung. So that's why it is more difficult to treat. And this graph you can see, that is people studied it in the patient. So small cell, although there are several treatment strategies like chemotherapy, radiation therapy, immunotherapy, sometimes some surgery, but the 2-year survival is about 10% and the 5-year survival is less than 5%. So the when somebody is diagnosed with small cell lung cancer, so you can see they are almost destined to death with the current traditional treatment. Actually, with that treatment, we are killing. I mean, radiation and chemotherapy, whatever you think about, we are using to kill the cancerous cell. And to kill means it's not like that you just put a bleach on and kill it, not like that, but we initiate the programmed cell death, the p53-dependent apoptosis, which is a very controlled process. And that is already in normal physiology, we have it, because whenever we are having some mutation, DNA damage over the time with replication and lot of stuff, lot of stress, it can initiate. First of all, our cells will try to fix that problem. If they cannot fix it, that time they will initiate the p53 dependent apoptosis, which is cell suicide. And in medical terms, that is called cytotoxicity. Now, interesting point is, in this small cell lung cancer, the p53, see the 91% radiation therapy, they cannot initiate the apoptosis because the regulator is not there. So in that case, this treatment won't work. So it's nothing surprising. It is obviously. In the early onset, probably it will work, but eventually over the progression, they will delete and mutate those p53. So we need something, treatment opportunity, that we can terminate this cancerous growth or proliferation, or other word malignancy, which will be p53 independent. If p53 independent, there is no problem. p53 is there or not, it doesn't matter. So now I will talk about what actually the cancerous growth, I mean, cancer we are talking about, what is it? I mean, is normal cell is converting there. So the basic physiology, and with that how it is turned into a cancerous cell, I will just go with the simple cartoon here. So as we all know, there are stem cells we always talk about, because those are the cells are responsible for replenishing all cells we need. Every time, every day we are losing cells, they are dying, injury or something, and we need to replace it, otherwise we won't be alive. So by the way, it's almost like every day we need 330 billion cells to maintain our normal living. So the stem cell, the growth is very slow. The embryonic stem cell or normal tissue stem cell, where you see, there is a purpose why it is the stem cell replication is so low, minute multiplication. It may be in once in a month or once in 2 months, that kind of. The doubling from 1 cell to 2 cell, that replication will happen. Because every cell division, when we studied in our college days or even in the PhD time, so every replication, there is a chance of mutation. And stem cell has to be a normal condition that there is no mutation as much as possible. So that we will be, the physiology will be maintained. So always it stays in a very slow replication. And then there is a lineage progenitor cells, which is destined to be a differentiated mature cells. Some will be kidney cells, some will be, doesn't matter, the myeloid cell, whatever, blood cell, depending on the different transcription factor and regulator, it from there, the progenitor cell, it divides. And in this progenitor cell, there is an exponential growth, which is coupled with the differentiation. And then we get into the differentiated mature cells, which is the normal physiology. The cancer cell, the cancerous cell, it always happens when it's turned into in this one. Because if it is that slow process, it's no good, because by the time the cancerous cell will overload there, with normal age will be dying. So they need the exponential growth. So always it will be the lineage progenitor cells that is converted into the cancer cell. But if they differentiate the normally, so there is no cancerous growth, because the growth will stop. Then terminal differentiation, if there is something, the apoptosis, which this replication stops and it stays the same way, because we know the mature cell doesn't divide anymore. It has its own function, they will do that, but it doesn't. So cancer, what it does, the first part, this replication is doing, but they are stopping this differentiation and the apoptosis. So first, they do stop the differentiation. So it is not maturing. In the exponential growth phase is there. But there is some mutation or something that it can happen because of that, or changes. That's why the differentiation is not happening. So our system will detect that. So they will try to initiate the apoptosis. So what they do? They delete the p53. So it cannot be apoptosis or cell suicide. So they are staying in that way. So that is the cancer. And MEK amplification means MEK is the master regulator of replication. So amplification, proliferation, whatever you say. So that's why we get the cancerous growth. So now I will talk here, but what I mean to say, if we could fix here, like the differentiation, the first thing, the loss of function of lineage differentiation, what cancerous cell is doing in this progenitor cell, if we could fix this, then it will be the normal physiology. So we can stop the growth. So here I am talking about that. This is little complex. I will go slow, but if you have any question, just ask me. So in stem cell, or doesn't matter progenitor cell, every cell replication genes are always open. We know about the we are open and closed, but heterochromatin and euchromatin, that kind of term you can think of. So when a transcription starts, you need a open chromatin structure. Then transcription factor can bind, and then gene will be expressed. So replication genes always stay on. Housekeeping genes, they are always constitutively open. But the differentiation gene is not, because you need a particular number of cells to finally mature it. Otherwise, you will have the required number 100 cells, and maybe you will have 10 cells, and that will not do our purpose. So there is several times of replication, and then it goes for the differentiation. So, and there is signal that initiate that differentiation gene and the chromatin opening. the epigenetic regulation, and then differentiation gene gets activated. So in normal physiology, it does that way. The cancerous, I am talking about small cell lung cancer, that's why I'm telling, but most of the cancer you will know that the same way it works. So what it does, it doesn't allow to open this differentiation gene chromatin structure. So the transcription factor doesn't bind there. So no differentiation. And how they do it? Because the co-repressor that is keeping the chromatin structure closed. So if we can remove this co-repressor from there and co-activator can bind there, co-activator will prime the transcription factor binding over there, and it will go for the gene expression for differentiation. And that way we can re-establish the normal physiologic process. This is my outline of the talk, and now I will go for the our data. So now, as I told, that is neuroendocrine origin, the small cell lung cancer, because it's already people have checked it, and what is their origin, because it was so difficult to treat. If you don't know the all details about it, it will be hard. So people, several group, they have shown, and very nicely they have shown, that all these small cell lung cancer, they are neuroendocrine lineage origin, because of their expression of those transcription factor. Not only this is neuroendocrine origin, it is these transcription factors are absolutely necessary. If you delete this the cancer cell growth will be completely gone. So these are committed to neuroendocrine lineage, the mature cell, whatever it was, and then it stopped to get mature. So now we focused on that neuroendocrine lineage gene expression as well as the replication gene expression. So there I showed the ASCL1 and NEUROD1. There are two main transcription factors, key transcription factor that regulates the neuroendocrine differentiation, pulmonary neuroendocrine differentiation, I should mention that. And the MEEK, as I mentioned before, that is the replication gene expression regulator. So you can see in normal pulmonary neuroendocrine cells, both are expressed same way. So it is replication as well as differentiation. These two processes are coupled, so they get mature. But when in small cell lung cancer, you can see that MEEK target, that is replication, is the same way with normal pulmonary neuroendocrine cell, but these are repressed, this gene, which is the differentiation genes. So specifically that is repressed. So it's not getting mature. And this is the plot where I'm showing that in small cell lung cancer, the neuroendocrine differentiation gene and MEEK target gene in negative correlation, because when MEEK is high and the ASCL1 targeted genes those are low. So it is just replication and proliferation going on, no differentiation. Now, there are epigenetic marker that how the gene regulation it happens. If, as we all know, the methylation, acetylation, those are the regulator for the gene expression. So you can see that this is embryonic stem cell. Okay, so in embryonic stem cell, there won't be any differentiation. That is always in a replication mode, because you need all the time stem cell to replenish our other cells. So here ESL targeted genes, the H3K27 methylation, which is the repression mark, epigenetic repression mark, that in embryonic stem cell is high, whereas MYC targeted gene is those methylation is going down, replication is going on. But in small cell lung cancer, you can see in both cases, these are, MYC is same like the embryonic stem cell, but ASL targeted genes, those methylation is gone, which tells us that these are destined to neuroendocrine lineage cells. But interestingly, when we check for the CpG methylation, which also regulate the gene expression at the transcription start site, you see in small cell lung cancer, the ASL targeted genes are highly methylated CpG region is more compared to the embryonic stem cell. MYC is similar way. So it is specifically repressing the ASL targeted differentiation gene. So it is in progenitor cell, its amplification is going on because of this, the MYC, with the MYC, it gets amplified, but it's not differentiating. This is the overall pulmonary, that is only the ASL1 targeted I showed, and this is the pool of pulmonary neuroendocrine lineage genes, and it showed the similar effect. So that confirms our findings. So now we are showing the ASL targeted genes and the pulmonary neuroendocrine genes, and the CpG methylation is inversely proportional in the gene expression level. RNA-seq data, you can see that. Now the CpG, methylated CpG, that is, there is an enzyme DNA methyl transferase 1, which is writer means is methylate the CpG region, and TET2 another enzyme that is delete that methylated part, and that time the gene expression happen. And you can see in small cell lung cancer the DNMT1 is highly amplified at the same time that opposes the DNMT1 function. TET2 is repressed. So recurrently deleted, I should say, the term wise, that amplified and this is deleted. And these are our observation, and now we are trying to remove or how to knock down this DNMT1 from this cancerous cell, or want to check that if we can restrict the small cell lung cancer proliferation. So to do that, we used two different human cell line for in vitro assay, and one murine cell line, which is p53 null cells, the F1339, and the human I will be mentioning for H82 and H146. And for in vivo assay, we use the mouse model, I made it, and the cell we use that one, the p53 and RB null, the small cell lung cancer that I just showed, F1339, to develop the small cell lung cancer. First of all, we check that if we knock down the, I wanted to see first, before knockdown, that whether we are claiming that, okay, DNMT1 is the one that is regulating the neuroendocrine gene expression and it is recruited over there. So ASL1 and NEUROD1, these are the key transcription factor that is regulated the differentiation gene. And our hypothesis is DNMT1, that is a co-repressor and binding to this transcription factor and not allowing it to express the differentiation gene. And to claim that one, we need to show that yes, it is happening. So we did co-immunoprecipitation assay as well as cut and tag. I will tell about this method. So you can see, like, in F1339 and H146, that ASCL1 and the NeuROD1, it is co-immunoprecipitated with the DNMT1. So they are interacting. And cut and tag is a like similar kind of thing like ChIP-seq. But here what we do, like we use antibody, specific antibody, and we use the live cells. So in the antibody will bind to that particular protein. Here we use the DNMT1 antibody, and it will bind to the DNMT1 wherever it is binding in the chromatin region, and where it binds, and immediately they cut the piece of the chromatin. And we pull that chromatin and we go give it for sequencing. And that way we'll know in which particular area of the chromatin the DNMT1 binds. And we did see the ASCL and NeuROD1 gene that is binding, the DNMT1 is binding there. So it confirms that DNMT1 is binding in the transcription factor, the neuroendocrine differentiation transcription factor, and it is inhibiting that way. So now we use the knockdown using DNMT1 siRNA, and you can see this is DNMT1 is knocked down in both cells, and then we showed the cell growth is inhibited in both of the cells. And we also checked for that gene expression. So there are the differentiation gene marker for the pulmonary neuroendocrine lineage. We used several, but we are showing like 4 genes here. So GRP, you can see that one, and NNAT, CHGB, and SCG2 or CHGC is same. They're called both way. So, and you can see in both human cell line that all the genes are highly expressed when we knock down the DNMT1. So it is inducing differentiation. And by imaging, we are showing the images. This is a Ginsa staining. And whenever the nucleus and cytoplasm ratio that increase, that is an indication of cell differentiation. It's starting. So you can see here this control, it is nicely round and the cytosol is less, and by DNMT1 the cytosol is more and the cell size is increasing. So that those are the indication of the differentiation. And we see in both cases up there. And this is the apoptosis assay, because we showed, okay, it is different inhibition, growth inhibition, and then that is differentiation, but we need to check it whether it is doing any apoptosis or not. So you can see the control versus the DNMT1, so there is no change. And fluorozoline, this is a apoptosis inducer, a chemical. We put it in cell and positive as a positive control, and you can see here it is inducing in the end. So our DNMT1 depletion is not inducing apoptosis. So now we also checked for that the co-activator and co-repressor level in those 2 cells by RNA-seq, and then you can see like DNMT1, it is highly amplified. The TET2, which I said it opposes the DNMT1 function, it is deleted. And SMC4, another co-repressor, that is condensin, that also highly amplified. CHD4 and SMC3, which is which opposes this SMC4 function. No, SMC3 is another co-activator, sorry. So that is also amplified, and CHD4 that also amplified here. And this one, the SMARCA CC1, I will show later. Those are, it is in H82 is very low, it's comparatively low level. HDAC, there are highly increased, but there are some HDAC inhibitor too. I won't say that there is not. And these are the other co-activators that is deleted. So overall, what I want to say, like the co-repressor that is highly amplified, and the co-activator is getting deleted from these cells. And first, I will talk about the DNMT1, and then I will be talking about SMC4 targeting. And our new project is going on CHD. I will show a little bit of data from there. So DNMT1, that enzyme can be inhibited by two FDA-approved drugs, that is decitabine and 5-azacitidine. These are prodrugs which target the DNMT1, and it is already people are using in the doctors are using for MDS and AML patient, like the acute myeloid leukemia and the myeloid dysplastic syndrome. So those drugs are people using for treating. And also teriflunomide, it's not for the cancer treatment, but multiple sclerosis people are doctors are using for ages. And why I am just mentioning these three drugs, not the others, because these three drugs are the non-cytotoxic drug that can work on pan cancer, not others. There are several other drugs, but not cytotoxic. That's why those are not our interest. Our goal is to make it non-cytotoxic drug, so patient will be healthy, no side effect, no healthy cell will die, so treatment will be better. And this is our own drug. We are developing this one, so it's ongoing work. So here I am showing that we targeted the DNMT1 by using the decitabine and 5-azacitidine, just I mentioned. So you can see compared to the control, it is going down. Araci, that is a chemotherapy drug, or it is other name is the cytarabine. And you see there is no depletion of the DNMT1. And I also showed in other two human cell, you can see so nicely it is depleted. These are murine cells though, that with the decitabine and 5-aza. And I showed the cell growth that is inhibited. So the blue line is the 5-azacitidine, red line is the decitabine. And decitabine and 5-aza, I didn't show the structure here, it will be too complicated, but these are analog of deoxycytidine and cytidine. So deoxycytidine and decitabine, those are analog, and azacitidine and cytidine is there. So we are actually getting competed out these two because this is a normal physiology. Whenever we are doing the DNA synthesis is happening, these are needed. So we are just replacing that with the decitabine product of that and the 5-aza for the cytidine. So we showed only the decitabine and 5-aza inhibited cell growth, not the deoxycytidine and cytidine, the same concentration we used. Same way, we showed the decitabine 5-aza also inhibited the cell growth in these two human cells. We showed the gene expression by qPCR, and you can see all these cells showed that all differentiation genes are highly expressed when we depleted DNMT1 by this drug. And there are some protein that expresses that marker for the differentiation. Differentiation is basically when it is exiting cell cycle. Normal replication is going on and then it exits cell cycle and gets mature. So cell cycle exit, usually the MYC and P27 will be higher and the MYC will go down, and you can see there is an indication of the going down, and the P27 is getting high. Different cell, they expresses different types of MYC. So this was CMYC and then LMYC. These cells also I didn't show it here. So the LMYC is highly expressed than the CMYC, and the regulation was nice. Then the same way as I mentioned before, we did the GIM sustaining to show the cells, and also by flow cytometry, these forward scattered and side scattered. Forward scattered is the cell size, and the side scattered is the cell complexity. So when cells get differentiated, cell size increases and also the complexity increases. So, and when it shifted from left to right, that means it's increased. And you can see that the cytabine, which the drug I am talking about, it is inhibiting the cell growth by depleting DNMT1. It indeed going for the differentiation. And also by RNA-seq data, we showed that the cytabine in these two different cell lines, that neuroendocrine genes, targeted genes that are amplified, and in both cases, whereas the MYC targeted genes are it's repressed in both cases. Now we showed that whether it is doing apoptosis, because we don't know that we are using the drug, so if it is initiating apoptosis also or not. You can see here these cells I am showing, that this is the vehicle means control, and this is the cytabine 5-ASA, and this is our positive control. So this quadrant, the annexin V will be highly expressed when it is going for the apoptosis. This is early apoptosis marker, and you can see these vehicle and treated groups is similar, whereas positive control, it is showing the increase of the apoptosis, which is we expected. And this one also, these other two cells also did the same way. So positive control showed apoptosis, not the drug. So it is non-cytotoxic. Now question is, all these are in vitro, whether it is working in the mouse or not in vivo. So we just first checked in the animal model that which is the standard chemotherapy drug, cisplatin and etoposide and vehicle, and you can see with the images that there is no indication of reduced tumor growth over there. And also the survival of the mice, it didn't change when we use the chemotherapy drug. So chemotherapy doesn't work because the cells we use, those are p53 null, it won't work. So now we checked for the DNMT1 targeting. So to do that, we use this mouse, and DNMT1 that we use decitabine, but they are decitabine or 5-ASA. But there is a standard treatment for small cell lung cancer, that is the immunotherapy. So anti-PD doesn't do much also, as you saw in the survival. So, and with that, we added DNMT1 plus inhibitor plus anti-PD if it improves. But you can see that DNMT1 alone and then DNMT1 and anti-PD doesn't have much difference. And anti-PD doesn't work that great. That is the immunotherapy. And these are the liver, because it's always metastasized in the liver from the small cell lung cancer, and this is very aggressive cells, so it does in the liver. So we showed by the liver weight as well as liver picture and also the section, and you can see that DNMT1 targeting, it reduces the cancer burden, the same as with the combination. Our only immunotherapy didn't do much. It's same, almost close to the vehicle group. And this is the without cancer, the normal mouse liver. And with this images also, you can see the tumor cell infiltration in vehicle group and DNMT1 inhibition, and the combination, it shows less entry, whereas and I should say immunotherapy that also has significant amount of cancerous cell infiltration. Now, we also did the same way that for the survival of the mouse. So this is, you can see the vehicle DNMT1 alone, and then immunotherapy plus DNMT1 targeting together. So you can see there that anti-PD didn't do much. DNMT1, it did inhibit nicely, significantly. Although it showed the combination is inhibition is there, survival, but it's not significant. So basically, it didn't improve much with the combination. So we wanted to check why it didn't. The anti-PD and immunotherapy failed. So we checked for the different immune checkpoint marker, and you can see the immunotherapy actually that increase. So that's one of the failure of that reason for the failure. You will see all over all those markers. There's other, we are targeting anti-PD PDL1, but its other marker is getting up. So that's why it is failing. And another reason might be the MSC class 1. So the MSC class 1 you required for the immunotherapy to act on. And in these cells, the small cell lung cancer, the base level of this expression of this MSC1 is very low. So that might be another reason. You can see here, compared to non-small cell lung cancer, the expression level is so low. Although the decitabine and 5-ASA, it increased a bit, but if basal level is too low, it's very hard to get that significant effect, I would say. Probably it will improve a bit, but not much. So now we thought, okay, if that combination didn't work, how we can improve that treatment with the other combination. So as I mentioned that there was a other co-repressor SMC4 condensing, which is required for that nucleosome, that open chromatin. So that one is highly amplified in these cells, small cell lung cancer, and the SMC3 is going low. So now we can target this SMC4 by using teriflunomide, another drug, which already people have used those for different reasons, but we used in the cancer cell. And you can see here that it indeed inhibited the cell growth of these cancerous cells. Also, it increased the gene expression in all those cells, and it did not induce any apoptosis. This is our positive control. And also all these cells, they induce the differentiation, which I am showing by the images. Now, we wanted to combine these two, DNMT1 inhibition as well as SMC4 inhibition by using the teriflunomide. And we did the survival assay, and you can see we meant the DH inhibitor. Why I'm telling, but I need to tell about that. So DNMT1, that one is targeting the deoxycytidine, I said, the deoxycytidine, the last one, which will incorporate in the DNA. And DH is an important enzyme that is pyrimidine metabolism. So in vivo there is a synthesis. So when I will try to compete out, so our body mechanism, it will be to increase the pool of the regular deoxycytidine. So that this
Madam, how many slides left? और कितने स्लाइड हैं?
ओ टाइम हो गया?
हो गया
Sorry.
5. कोई नहीं करिए ना. कितना है, नहीं तो दूसरा लेक्चर करवाएंगे.
ना सॉरी, रुको. ओ कहाँ चला गया?
बंद कर दिया हमने.
ना जस्ट 3-4 slides.
ठीक है फाइन.
Okay.
यस.
So this combination, you can see like that DNMT1 targeting it inhibited as I showed before, and also the teriflunomide the same way extend, but when we use the combination, it extended significantly. So it indeed improved the treatment, and we repeated again and we saw the same effect. And this is the liver weight. You can see how significantly reduced the cancer burden, and there is no problem in the blood profile. So it improved. That combination is better than the single drug. So our for this part of my work, I will say like DNMT1 and condensate targeting activate terminal differentiation in chemorefractory small cell lung cancer. DNMT1 and condensate targeting decrease chemorefractory small cell lung cancer burden in vivo, and combination treatment is better and significantly extended survival of compared to the single drug therapy. And this will be my future direction. I won't talk about. I had few slides, but that is okay because this is ongoing process. So I will go to my acknowledgement. So I want to thank my PI, Dr. Sunthara Raja, for giving me opportunity to working on this interesting project, and all my current lab members and previous, and all our collaborators, and for funding all these funding agencies. And thank you all.